screen and start their pitch now. Thank you.
Okay.
The first, yeah. Okay, you may take the stage now. Thank you.
You can see my screen, right?
Yes, we could.
Okay, yeah, so I'll start. Good afternoon, everyone. Today we'll be sharing our project on medication history and age-related brain morphology in children and adolescents with ADHD. So these are the things we'll be covering today. Starting with a brief introduction, ADHD is a neurodevelopmental disorder that has been linked to delayed or atypical brain maturation, and structural MRI studies have reported abnormalities in gray matter volume, cortical thickness, and subcortical regions. So these differences are found in ADHD relevant areas such as the prefrontal cortex, anterior cingulate cortex, basal ganglia, cerebellum, insula, and reward-related regions. And the maturation delay model has been widely used to explain ADHD, proposing that brain development in individuals with ADHD follows a delayed version of typical development. However, this model does not fully explain ADHD heterogeneity because brain structure may differ depending on age, symptom course, ADHD presentation, and medication exposure. So this brings us to the research gaps. A study on psychostimulant medication found to normalize some ADHD-related structural abnormalities, especially in regions involving in attention, salience, and reward processing. However, many medication-related SMRI studies are small in sample size, ROI-based, and inconsistent across brain regions. Many studies have also compared medication and unmedicated groups directly, but fewer examine whether medication history changes the relationship between age and brain structure. The second gap is methodological. So most studies rely on one SMRI approach, such as BBM-derived gray matter volume or SBM-derived cortical thickness alone. Since gray matter volume and cortical thickness capture different aspects of brain morphology, it remains unclear whether combining BBM and SBM can better detect age-related medical structural patterns. I'll pass the time to Nandini now.
So the aim of our study, the primary aim of our study is to determine whether medication history moderates age-related gray matter volume in pediatric ADHD students. We do this using a voxel-based morphometry framework. Then our secondary aim is to evaluate the incremental utility of SBM by combining thickness matrix with volume data and thus directly comparing whether a multimodal VBM plus SBM framework reveals a distinct age medication interaction that are missed by a standalone VBM analysis. Moving on to our research question. Our first research question deals with if medication history moderates age-related structural brain deviation in children and adolescents with ADHD. So our first hypothesis would be, will a masked univariate GLM will reveal a significant interaction between age and medication history. We expect to see results in subcortical gray matter such as left nucleus accumbens. Moving on to our second research question. Next slide, yeah. Will the inclusion of SBM matrix provide a significantly higher global model fit and better control over spatial variance as compared to a single modality volume analysis, and thus giving our hypothesis if the statistical model accounting for both voxel-based and surface-based should explain significantly more variance in age-related developmental pathways than a standalone VBM model. Passing my time to Grant to explain our data source and analysis methodology.
Thank you, Nandini. Okay, so we'll be drawing our data largely from this data source, which is the ADHD 200 repository. So the reason why we chose this is that it's one of the largest data sets currently available for ADHD research out there. And besides that, it has already been pre-processed in multiple manners, which enables us to directly jump into the VBM analysis without having to go through the step-by-step pre-processing. So one caveat is that although the data has been pre-processed, we do still have to do some additional steps given that the data comes from multiple different sources or sites. So therefore, there may be some scanner-related data factors that we have to consider in order to ensure that it is properly harmonized and ready for analysis later on. So moving on to our analysis, with regards to our first research question, we will be adopting, a look at how age and medication can both independently and through an interaction influence gray matter volume change in ADHD. In order to do this, we looked through our data already and we saw that there are many different variables that the data set actually collects for us in order for us to be able to control for them and therefore isolate the effects of age and medication. So these include gender, ADHD subtype, the handedness of the participants, as well as whether comorbid conditions are present. So we notice things like anxiety, depression, and ODD. So individuals with these comorbid conditions will be excluded. So moving on to the next slide. The way that we intend to do this will be through a GLM analysis as shared previously by Nandini. So in looking through the literature available on neuroimaging analysis, we found that one major thing that we have to consider is the possibility of false discovery, which can occur as a result of how VBM analysis is conducted, where we are running multiple, like quite a large volume of parallel comparisons between all the individual voxels within each brain. So to address this issue, we will be introducing the Benjamini-Hochberg false discovery rate correction in order to correct for this possibility of false discovery. So in order for the GLM to work, we propose this particular manner of looking at the DV where we control for sex, handedness, and ADHD symptoms in order to isolate the effects of age, medication history, whether the participant has taken medication or not, as well as the interaction of both these terms. One thing that we are looking at will be to whether or not we should include or exclude ADHD symptoms, given that this is already correlated with various other terms within our analysis, such as sex as well as age. Yeah. So moving on to the last part of our analysis, as shared by Nandini earlier, we also intend to look at how, whether VBM or voxel-based morphometry alone can provide us with a sufficiently large, a sufficiently in-depth explanation, or whether combining this with surface-based morphometry will provide us with a more in-depth look at the influence of age and medication in ADHD brain development. So in order to do this, we propose the use of z-scoring the values for both the SBM and VBM metrics, since they come from different data sets available within the ADHD 200 repository. Hopefully in doing so, then we will be able to appropriately compare them in our analysis. And this is given that we have enough time during this project. And with that, we've come to the end of our presentation. Thank you very much.
We'll pass it on to questions. Oh yeah.
Thanks presentation. Very quick and concise. I may have missed this. So how are you going to operationalize your ADHD symptom severity?
Within our data set, the data actually includes a few different scales for identifying the severity of ADHD. So this has allowed them to classify symptom severity in terms of inattentiveness as well as hyperactivity. So we hope to use this during our analysis.
Okay, so it's sort of standardized. So you have just two measures, right? At the end of the day? Okay.
Yep.
And then I think one of the things you did mention, but to clarify again, the inclusion-exclusion criteria later on, because you are excluding some of the subjects in the existing data set, right?
Yes, we will be.
Yeah. And also the age group is quite, the range is quite large, right? So you also be looking at that as a dimension, right?
Yes, I think on the next slide, we have it included. So we'll be controlling for things like sex and handedness, as well as ADHD symptoms. but the main thing we'll be looking at in terms of like effect will be whether age and medication history actually influences gray matter volume over the development. Yeah.
Okay. And you have already identified your subjects to be 478. Is that confirmed or not?
This is the approximate one we have. This is our based on initial screening of like excluding all the data which is missing or invalid. But we still have to take a closer look at where we have not excluded individuals with comorbid conditions yet from this number range.
Okay. Although this is an exercise looking at the methodology and the approaches that you use, but what could you conclude in this study? Like what would you like to find out about medication severity? Like what is it that you want to find out from the study that is not already known in the literature?
Okay, I think one major thing would be because when we look through the literature currently available, a lot of it is based on VBM. So a lot of the analysis going on in terms of how age or medication independently influences brain outcomes is based in VBM. So we hope that by looking at it with SBM as well as through an interaction lens, we will be able to better inform both of these views at once.
Yeah, but pushing it a little bit further, so what, if you found out that there's differences in the cortical thickness and all that, so what would that imply? You may want to think about, like, in the literature, what knowledge about that would be helpful. I think this is pushing it a little bit beyond the methodological questions that you've been asking. Okay, but nevertheless, good job with the presentation.
Hello, I have a quick question, just out of curiosity. Which programming languages do you plan to use?
I think from my research and up till now, we plan to use Python.
For the entire project, including the stats?
Yeah. Grant, do you have anything to add? I think from my understanding, we are mostly using Python, right?
Okay, cool. There's a Python library called stats models. If you're using GLMs, you can look it up. Just out of curiosity.
Okay,
Very
thank
interesting.
you. Yeah. Also, I think for Prof. Annabelle, which you were asking about, like the real life implication, I think the so what is just to prove like the importance of like using multimodal analysis as compared to like a single analysis, that maybe using a combined analysis tool of SBM and VBM will give us more clearer understanding of how medication history works. I think that was like what we wanted to take away or be like the real life implication.
Okay, that's a good near point. Yeah. You may want to think about the different sets, because what you propose actually is quite complex, but seems like you have stated out your different steps. You probably would be maybe doing some statistical analysis at the end. Python is good, but you could also consider R, maybe more straightforward with some of the analysis later on. But definitely good to look into the various steps, that existing software that for VBM and SVM that you could already use.
Yes, for sure. Thank you, Prof.
Thank you.
So thank you, Grant, Agawa, and Sunyi for the presentation. We'll move on to the next person, Clarice Leong. Can you just put your screen out and start presenting? Thank you.
Sorry, can you see my screen? Yes, right?
Yes, we can see your screen and we can hear you as well. Yep.
Okay, is it okay?
Yep, looks good.
Okay, I'll start. Okay, hi everyone, profs, TAs, friends from Taiwan and Singapore. My name is Clarice and I'm from NTU Singapore, and my project is titled Connectome-Based Predictive Modeling of Cognitive Performance in Psychiatric Disorders. So a patient walks into your clinic, he has been diagnosed with schizophrenia, you prescribe antipsychotics to manage his symptoms, but yet 6 months later, his psychosis is controlled, but he still cannot hold a job, follow instructions, or live independently. Why? Because antipsychotics mainly target psychosis and not cognition. Cognitive deficits like impaired working memory and attention persist even when symptoms are managed. And these are what actually determine whether a patient can function day to day. Despite decades of research, no neuroimaging biomarkers have been adopted in routine psychiatric clinical practice. This is not an isolated case. Mental disorders affect over 1 billion people worldwide, accounting for 16% of global disease burden and $5 trillion in economic losses annually. Yet, diagnosis still relies mostly on symptom-based criteria with no objective biological measures to guide treatment. There's currently no way to predict which patients will develop cognitive deficits or how severe they will be. Thus, my project aims to determine whether resting state functional connectivity can predict individual cognitive performance transdiagnostically across schizophrenia, bipolar disorder, ADHD, and healthy controls. To answer that, I need to explain what I'm measuring. Resting state fMRI scans the brain while a person is doing nothing at all. I use resting state specifically because cognitive deficits in psychiatric disorders are persistent and not necessarily task dependent all the time, which means that resting state fMRI is more appropriate for studying them than task-based fMRI. From this scan, I built a connectome, which is a map of how brain regions communicate with each other at rest. Each person's connectome is unique to them, like a fingerprint. And by taking a transdiagnostic approach with my dataset, I study what schizophrenia, bipolar disorder, and ADHD share at the brain level rather than treating them as completely separate conditions. The tool I use is Connectome-Based Predictive Modeling, or CPM, which has been quite popular, and I saw that there has been relatively a lot of publications recently. Looking at this figure, each participant has a connectivity matrix from their resting state scan and a cognitive score, which is the phenotype that we're investigating. I correlate every brain connection with cognitive score across participants, keeping only the most significantly correlated ones, sum those connections into a single value per participant, fit a linear model, and apply that model to predict a held-up participant score. So this works by the leave one subject out cross-validation. Yeah, so the red figure on the right is the person that the model has never seen. I repeat this for every single participant, and this is what makes CPM different from most neuroimaging study. It predicts at the individual level rather than comparing group averages, making it clinically meaningful. Prior work on this dataset only has ever looked at one domain at a time, and nobody has applied resting state CPM across 5 different domains simultaneously, cognitive-wise. And that's the gap that my project hopes to address. As for my dataset, I'll be using the UCLA Consortium for Neuropsychiatric Phenomics dataset, which is publicly available on OpenFMRI, with 247 participants across 4 diagnostic groups. I target 5 cognitive domains consistently, impaired across all 3 disorders: working memory, attention, verbal memory, processing speed, and inhibitory control, each measured using validated neuropsychological tests from the dataset itself. This directly applies the BrainHack School modules, functional connectivity in fMRI, machine learning, open data, and brain imaging data structure, as well as Python and R, which I hope to use R for my project, data visualization, and introduction to Git and GitHub. To conclude, if resting state connectivity can predict cognition across disorders, then it changes how we think about clinical assessment. Rather than running a full neuropsychological battery such as the Monreal Cognitive Assessment, a single non-invasive resting state scan could one day potentially tell a clinician which cognitive systems are at risk in a patient regardless of their diagnosis. This project is also a step towards precision psychiatry, where treatment is guided by an individual's brain and not necessarily just their diagnosis. Thank you. These are my citations.
Hello. Can I
Hi.
ask like 2 questions? By a linear model, do you mean logistic regression?
Yes. Yeah, but that's towards the end. So it's comprised in the whole CPM model. Yeah, so firstly, I'll run the leave one subject out cross validation. So each time I'll have 5 participants, 4 of them will be trained on the model, and then we'll apply this model onto like the person that is not trained. And then I run this across the 247 participants.
Okay, and sorry, I might have missed it. So you're planning to add other metrics like working memory into the as features into the model as well,
Yes.
along with the connectivity matrix.
Correct. So it will be the resting state functional connectivity and also the behavioral data within those cognitive domains that was measured by the different neuropsychological assessments that they did.
Okay, and last question. I'm a bit pessimistic, but what if your model underfits? What if it doesn't pan out?
Actually, I kind of ran some preliminary analysis already. So, if I remember correctly, I think working memory and attention was the one that gave the most significant results. But there were some that were non-significant. But I think that too is like a finding in itself. Yeah, but I get what you mean. Cause
Okay.
yeah, at this stage, like I also haven't controlled. Like I'm planning to partially control for sex, age, and also education years. Yeah, but I haven't done that yet for my preliminary analysis. So that is something that I will work on. Yeah, thank you for your question, though.
Okay. Yeah, very interesting. I look forward to your results.
Thank you.
Nice presentation, Clarissa. Did you share the demographics of your population sample again?
Here, this one.
Okay.
So I actually took out these groups, the schizophrenia, bipolar disorder, ADHD, and healthy controls.
So they are collected across, so it's part of a consortium, so you're collected across different sites?
Yes, from the UCLA dataset.
Okay, and so they're already harmonized by the time you do the analysis?
Correct. So I'll have to pre-process it also.
Okay.
Yeah, but there is already the dataset itself that was on the website had already had a pre-processing pipeline. Yeah.
All right. So you're following all that.
Correct. Yeah, and then the CPM code is also widely available in GitHub by people who have presented this before. Yeah, for example, the Shen et al. Citation here, I think he was the pioneering paper for CPM and functional connectivity.
Okay, good. Thank you.
Thank you.
Sorry, this may be a very, very naive question. I wonder whether in this consortium, there are records about their medication. I don't know, like what kind of medical treatments they are undergoing, and whether that might affect the connectivity also in some way.
Thank you for your question. That's true. I think I haven't looked into deeply to that because I focused more on the behavioral phenotypes in the cognitive impairment sense. So I've only seen the behavioral data for the cognitive assessments that they ran, and I have not yet checked the medications, but that is something that I can look into. Yeah, thank you for mentioning that.
I think there's some hands up. Just open up, turn on your microphone and ask.
Actually, students can ask questions too, not just the instructors
Okay.
and TAs.
So, wait, those were past questions.
Yes, Prof.
Hi, Clarice.
Hello.
Yeah, great presentation. Yeah, I have a question about interpretation. So you're trying to predict across five cognitive measures, right? Or
Correct.
cognitive
Wait, let
domains.
me go to the slide.
So and so how will you use these five to conclude whether whether resting state functional connectivity would be a good replacement for all of these neuropsychological assessments? Because that's what you're trying to achieve, right? I guess having a single resting state scan to replace these measures. So would it be like 3 out of 5, or what's your criteria to make that kind of interpretation?
That's a good question. I think I haven't really thought about that, but I think the lens that I was going to also was like perhaps like if we manage to find at least one cognitive domain even, then that would be a good sign that it is something that can be like a form of triangulation, like when we come to see cognitive deficits and also different psychiatric diagnosis as a whole. Yeah, so for now actually if I remember correctly, 2 out of 5. So just now I mentioned working memory and attention was actually significant. Yeah, so I think personally I believe that's a good start. Yeah, and also why these 5 cognitive domains, right? It was because these 5 cognitive domains were common across the different psychiatric diagnoses that I included here. So schizophrenia, bipolar disorder, and ADHD. Yeah.
Okay, great. Thank you for clarification.
Thank you.
Thank you very much, everyone, for the presentations and the question and answers. So just to regulate a bit of timekeeping, we started a bit later as well due to some technical issues. But I think now is a nice time to cut over to the talk with Dr. Jeremy first. And then we will continue with the pictures afterwards as well. And for that, I will just pass over to Josh.
Yeah, thank you. Thanks, Yongsheng, and thanks to the presenters so far. And Jeremy, are you online? Can you...
Yes. Hi, Joshua. Hi, hello, everyone.
Okay. And I just make an informal setup first, introduce you informally to everyone while the people busy with the technical setup. I don't know if you all need to record or whatever, or stop the recording. You mean to stop? I think I gave you host control. Yeah. So okay, so yeah, and Jeremy, in case you don't know, there's Singaporeans and Taiwanese all here in this session. And we have from the Taiwan side, myself, and you have Charlene over there from linguistics department. And then actually over here we have Kevin Junxian, who is behind me, who is trying to hide so you can't see him. But he's at Zhongyang there. Okay, and then over at Singapore side, we have Annabelle, who at the psychology department, Nanyang Tech, and then also Darren is there as well, also from